Hello, and welcome to Chapter 9, where we are going to talk about genes and inheritance. When we talk about genes and inheritance of genetic traits, maybe you immediately think of your family resemblance. So, how you look like maybe your mother, your father, or your grandparents, or maybe even your siblings if you have any. The reason that we look like our family members is because we have this genetic makeup that matches many of the genes that we share with our family. You inherit your genetic makeup from your parents. You get half of your genetic information from your mom and half of your genetic information from your dad. We have 23 pairs of chromosomes. One is maternal, meaning your mom, one is paternal, meaning your dad. And gentle reminder of when it comes to things like genes, we are going to have two, technically, versions of a gene. These are known as alleles that come from your mom, one from your dad, and that is going to lead to something called a genotype. So, when we look at genes on a chromosome, remember you have two sets of chromosomes. One set is from your mom, one set is from your dad. So on those chromosomes, we have our genes. You're going to have half your genetic information coming from your mother and half from your father. So when we talk about inheritance patterns, this is going to help us understand and predict certain traits and behaviors, things that are passed from parent to offspring. What we call this passing of traits from parent to offspring is heredity. There are quite a few traits when it comes to humans that can be controlled by a single gene. However, again, humans are a little bit on the complex side. We do see quite a few traits that are controlled by multiple genes. We'll talk about that a little bit more later. Now, speaking of essentially controlling genes, we are looking at this ability to manipulate heredity through something called selective breeding. For example, this very tiny little horse. That is a product of humans manipulating the breeding process in order to breed out traits that we do not want and to selectively breed for traits that we do want, leading to something like this tiny horse. When it comes to a handful of traits, though, in humans, we do have single gene traits, meaning one gene for one trait. However, there are not that many, just a handful. When it comes to something like a cat, what is a really nice example of a single gene trait is going to be something like fur length and fur color. These are both traits that are controlled by a single gene that is passed from parent to offspring when it comes to making cats. Now, a handful of characteristics can be influenced by multiple genes as well as the environment. We will get to that in a little bit. So, what do parents give their offspring that makes us have this similarity to our parents? Well, initially, there was kind of a mistaken idea that in each sperm cell, a tiny, fully formed human being existed. And of course, this was not a correct way of thinking, and that was that leading idea that was popular through the 1800s. Now, I need to introduce you to someone very special. That is Gregor Mendel. He is known as the father of genealogy, or the father of genetics, essentially. And he did a lot of experimentation with pea plants in order to answer this question of what do parents give their offspring that makes it to where the offspring looks quite a bit like the parent. Now, with this odd idea that in each sperm cell there's a fully formed human being, that would make sense if we look just like our fathers, but we also have traits that look like mom. So this idea, obviously, which is not true, not correct in any capacity, is especially not true and doesn't help support why we would look like our parents. So let's talk a little bit more about Gregor Mendel's experimentation and how he really supported the modern idea of this inheritance of genetics from both parents and not just one. Gregor Mendel focused primarily on pea plants. He was a monk and a botanist, and he had a garden that he could work on. And pea plants are a really fantastic organism to study for a handful of reasons. They're easy to maintain, easy to breed, and they reproduce pretty quickly. Likewise, they also have traits that can be easily categorized, such as flower color, pea shape, and the pea color. And he was able to look at the variation of these traits to determine whether or not he had true breeding, is what he called them, purple flowered plants, or true breeding pea green plants, things like that. However, a primary focus was looking at the flower color. So what he established were these distinct breeding populations. He would breed two purple flowering plants together, and what he would get is all offspring that were purple flowering. Likewise, he also would breed the white flowering plants together, and he would get true breeding white flowering plants. Now, he called these true breeding because he knew that the outcome would always be what the parents were. So then he started getting a little bit curious what would happen if he bred a true breeding purple flowering plant with a true breeding white flowering plant. So what he discovered in breeding these two plants together, the true breeding purple and white flowering plant, is that all of the offspring ended up purple. But he knew he had bred that white flowering plant with the purple, so why wasn't he getting any white flowers? If he took the no longer true breeding flowering plants and he bred them together, what he noticed is he got mostly purple flowering plants as well as a handful of white flowering plants. Now, this should look very similar to the lab that we did with the corn. So we took two cobs of corn. One was purple and the other was yellow. And we established that one of those traits was dominant and the other was recessive. That is exactly what Mendel was doing here. He ended up observing that the purple flowering plant is the dominant trait, while the white flowering plant was recessive. Now, when he bred the two true breeding plants, he realized that the recessive trait was being masked by the dominant trait. The recessive trait was still there, it was just not showing itself. And that is kind of the definition of our dominant versus recessive traits. Now, one of the things I want to establish here is that Mendel didn't really know what a gamete was, so a sperm versus an egg, and the fact that they were haploid, so had half the number of chromosomes, and that during fertilization, gametes will fuse that lead to a cell that will make that offspring, and that offspring is going to start as a diploid cell with two copies of each gene. But he still came to this conclusion, essentially, just not knowing how he came to this conclusion. There are three important observations from Mendel's research that we still use today. The first is that each parent puts a single set of genes into each sperm or egg that it makes. We know now that this is essentially describing the process of meiosis. So that process of making sperm and egg cells, those gametes, that have half the number of information from the originating parent cell. Next, we have offspring will receive 2 copies of the instructions for any trait, and that is describing a genotype. Remember, a genotype is made up of 2 alleles. One allele is from the mother, one allele is from the father, and together we have those 2 pieces of information that lead to the genotype, which leads to that physical manifestation, that phenotype, which is that trait. And then last, the observed trait in an individual depends on the 2 copies of the gene that it inherits from its parents. And this is that new term that we just were taught, that is heredity, that passing of traits from parent to offspring, which is why we end up looking much like our parents. Gentle reminder of the definitions for a phenotype and a genotype. A genotype is going to be that genetic information, so that is found on your DNA, that codes for RNA, that codes then for a protein, and that physical thing, the protein itself, is the phenotype, that is the outward appearance or the physical display. For genotype combinations, there are 3 of them. Gentle reminder that for each genotype, you must have 2 alleles. And remember, alleles are just variations of a gene. So we have the first combination, which is going to be a homozygous dominant genotype. This is going to be made up of 2 dominant alleles, and the individuals first, the phenotype goes, will display the dominant condition. A homozygous recessive genotype is going to be 2 recessive alleles, and the display will be the recessive condition. The third genotype combination is heterozygous. This is where we have 1 dominant and 1 recessive allele together, and because the dominant allele will mask the recessive allele, we end up seeing the dominant trait being displayed. Okay, so now we're looking at Punnett squares. This is how we are able to look at the probability of 2 individuals having offspring of a particular genotype. So what we are seeing here, I'm going to do my best to mark this out with a little marker here, is we see the genotype of a mother and then the genotype of a father. And we are going to set this up in a square pattern where we are separating, and I meant to grab a different color there, separating out the possible alleles that the parent can pass on. Now, when we look at 2 alleles together, remember that that is a genotype, so we can see the genotype of the mother and the genotype of the father. However, remember that the mom and the dad can only pass on one of these 2 alleles. So only one of them is going to be passed on, and we are looking at the probability of which one they pass on, and compared to the other parent, what is the combination that we're going to end up getting? So when we look at the individual allele by itself, this is known as a gamete. So this is the gamete that mom can pass on. This is the other gamete that mom can pass on. This is the gamete dad can pass on. And this is the other gamete dad can pass on. But they're only going to pass on one. So what we can see in the middle here, this combination situation, is that if mom passes on this recessive allele, then dad can potentially pass on this recessive allele, and the offspring will end up having this potential genotype. And we can see this for all the possible combinations. So we have 4 possible combinations of genotypes for the offspring based on the parents' genotypes. Now, in this instance, because mom is homozygous recessive, So she has two recessive alleles, and dad is homozygous dominant. That means he has two dominant alleles. The only option that their offspring will have as a genotype is heterozygous. Okay, this is one of the patterns that we can follow when we cross a homozygous recessive individual and a homozygous dominant individual, compared to a heterozygous individual being crossed with another heterozygous individual. This is going to give us a little bit more of an option. So if this dominant allele is passed on from this parent, and this dominant allele is passed on from this parent, we're going to end up with a 25% chance that the offspring is going to be homozygous dominant. If this parent passes on its recessive allele and this parent passes on its dominant allele, we are going to have this option, which is heterozygous. Now, we actually have this happen twice because we have the same thing happening over here where this dominant allele is being passed on and this recessive allele is being passed on from this parent. We end up with a 50% chance that the offspring is going to be heterozygous. We also have the potential of this recessive allele being passed on from this parent and this recessive allele being passed on from this parent, which is going to lead to a 25% chance that the offspring is going to be homozygous recessive. Please make sure you know how to set up and use a Punnett square. That is absolutely going to be on the exam. We could set up Punnett squares all day long, essentially, and get those probabilities of what are the chances that the offspring is going to be that genotype. However, a lot of what happens, which sperm is going to be created versus which egg is going to be created, and then which sperm is going to fertilize which egg, is all up to chance. Okay, so it is all about chance. However, we can predict those probabilities, but in the end, we can't really determine what exactly perfectly is going to happen. Because again, it all comes down to chance. Now, let's say that what we want to do is look at the potential of the offspring that we can get from two parents, but we only know the physical features that tell us a little bit about the genotypes of the parents that we are wanting to mate. So in this example, we have white alligators, and we are going to perform something called a test cross. A test cross is going to help us determine what is the probability or chances that this parent has this particular genotype and what is the offspring going to look like. So in this example, the only parent that we know for sure what their genotype is the mom. She has a physical feature, a phenotype that tells us that she has two recessive alleles. So she is a white alligator, and we know that in order to be a white alligator, you have to have two recessive alleles. So we know the only thing that mom can pass on is that recessive allele. The dad is the one that we are not quite sure what his genotype is. All we know is he is normal pigmented. That means he has to have one dominant allele for sure. What we don't know is what is his second allele. Is he homozygous dominant or is he heterozygous and a carrier for the recessive condition? So what we end up doing in this test cross is we set it up in such a way that shows us if he is homozygous dominant, we are going to see all of the offspring look like him. They are all going to be pigmented because they will receive that dominant allele from dad. However, there is the potential that dad is heterozygous, and that will give us these results, where we end up seeing the offspring be potentially 50% heterozygous, but they will look like dad, right? Because they will be pigmented. And 50% will inherit that recessive allele if he is heterozygous, and they will be white like mom. So this is how we set up a test cross. It is a way for us to look at the potential genotype of one of the parents when we're not quite sure what the genotype might be, if they are homozygous dominant or heterozygous. That leads us into a pedigree. Pedigrees are very useful tools that help us look at a trait and its inheritance within a family across multiple generations. So all I need you to know about a pedigree is that it is going to help us determine whether a trait is dominant or recessive, sex-linked, or autosomal. And remember, we will combine those terms to where it is dominant sex-linked, dominant autosomal, recessive sex-linked, or recessive autosomal. Now, when we talk about these terms, sex-linked and autosomal, we are referring to the chromosome. If you have a sex-linked trait, that is going to be found on the X chromosome. Now remember, for biological females, that is going to be XX, and this is going to lead to a genotype that is pretty much exactly what we've been talking about this whole time. So maybe if the allele was an A, we would represent it as an A. We would end up putting X and then the allele if it is a dominant, and a recessive if it is a heterozygous genotype. Now, males are slightly different when it comes to sex-linked traits because they only have one X chromosome. Their other chromosome is a Y. That means that there is only one option on the X chromosome for them to receive a dominant, ooh, that was an ugly A, or a recessive allele. So they only get one option. They don't get a normal genotype. When it comes to autosomal genotypes, that is going to be just like a normal, how we've written it before, the two letters together, a dominant and a recessive, or a dominant and a dominant, or a recessive and a recessive. But this is going to be found on the 22 chromosomes. Oop, I'm going to write a 22 on here. 22 chromosomes versus the sex-linked, which is only found on that 23rd chromosome, which is the sex chromosomes. Now, when it comes to the pedigree, I don't need you to know how to do a pedigree puzzle like we've done in lab, but I do need you to know, what do the circles mean? What do the squares mean? And what does a line that is connecting a circle and a square mean? What does it mean when there is a line coming down and connecting these circles and squares? So what we can see here, let me get a different color, is when we have a circle and a square that has a line connecting it, that is a mated pair, versus this line that is connecting them coming down from a mated pair, these are siblings. All right, so a couple of terms that we need to know involve kind of odd situations that we can see when it comes to genetics. These are not crazy frequent, and in fact, I only have a handful of examples that I do want you to know. For this first term, it is incomplete dominance. This is a condition where we see a phenotype of a, and make sure you circle, highlight, and underline it, a heterozygous individual. So this is going to be somebody who has some type of condition where their alleles do not match, right? So we have a dominant and a recessive, something like that. They are heterozygous. They are not homozygous, which would be something like big A, or little a, right? Sorry, my A's are ugly. I am drawing and writing with a mouse, so don't judge me too harshly. So this individual is heterozygous. And instead of having a dominant trait that takes over, instead we see a situation that is the blending of the traits. Let me give you an example. Snapdragons are a really cool example of incomplete dominance. This is where we have an allele that is called C, and it's going to have kind of an extra piece of information attached to it where we have the indication of color. So CW is going to be the allele that leads to white flowers for a snapdragon versus CR, that is the allele that is going to lead to red flowers on a snapdragon. When we combine these alleles, we get CWCR, and because this is an example of incomplete dominance, we do not see a red or a white snapdragon. We see the blending of the traits, which is going to lead to pink snapdragons, which, if you know colors, if you combine red and white, you get pink. Now, what's kind of interesting, if we take two snapdragons that are pink, so they have the genotype CWCR and CWCR for the other parent, they are both pink snapdragons, we are going to end up with this neat combination of getting some white snapdragons, some red snapdragons, and also potentially 50% pink snapdragons. So essentially what combining two heterozygous individuals when it comes to incomplete dominance, you can unblend the traits, which is super neat. Okay, moving on to another weird condition. This is again going to be a condition that is happening to a heterozygous individual where they have two different alleles. And instead of a blending of the traits, we end up seeing both traits coming out together. So this is called co-dominance. And I have a few examples for you to write down. The first is this black and white chicken. This is if we combine a black feathered chicken with a white feathered chicken, their offspring will have both of those traits. So individual black feathers and individual white feathers. Another example of codominance comes in the form of our blood type. Actually, it is a specific one of our blood types. Not all of our blood types are going to be codominant. It just has to do with one of the blood types. Now, this should look a little familiar from lab. We have three alleles, IA, IB, and lowercase I, that leads to 6 genotypes depending on how you combine the alleles. IA, and IA lowercase I will lead to type A blood, which is a phenotype. IB lowercase I will lead to type B blood, and IAIB is that example of codominance that leads to AB blood because we see both of those traits coming out at the same time. And then we have lowercase I, which leads to type O blood. Now, what does all of this mean? When we look at the phenotype of blood, that physical display, there is a physical structure that is caused from those alleles combining into the genotypes. For type A blood, you're going to have a physical structure called an A antigen. For type B blood, you have B antigens, and AB, you're going to have both A and B antigens. Antigens are going to be physical structures on the outside of the cell that are going to act kind of like a signpost. I like to think of it more like an ID that tells other cells in the body, kind of like your immune system, that, hey, those cells are meant to be there. They belong. Antibodies are going to be part of your immune system, and they're going to make sure that you don't have the wrong antigen in your body. So if you don't have the right antigen and you're a cell in a body, then these antibodies are going to kick them out. They're going to destroy that foreign cell. So, what do the antibodies look like? Well, for type A blood, it's going to be a B antibody because there's only one other type of antigen, which is a B antigen. So, these antibodies are going to be specific for B antigens. Whew, words are hard, guys. For B blood, type B blood, you have those B antigens, which means you're going to have A antibodies, because again, the only other antigen is A. When it comes to type AB blood, you have both antigens. That means you can't have either antibodies or you will attack your own cells. But for type O blood, what's interesting is you have neither antigen. So neither A or B. This means you can have both types of antibodies because your body does not want to have any of those antigens inside of it. This is an important piece of information when it comes to compatibility for blood transfusions. If you have type A blood, you can receive type A and type O because you need to match the antigen. If you are type B blood, you can receive type B and type O. If you're AB, you can receive any blood type because you have both antigens, so you can receive A, B, and O. If you're type O blood, then you can receive only O because you can't have any of the other antigens. But that does mean that if you're type O, you can give your blood to any of the other blood types. So the universal recipient is going to be AB positive. The universal donor is O negative. That positive and negative that we add to blood is just another antigen. And I don't make you guys have to know that, not really a big deal. Just know it's another form of essentially like an ID, kind of a way to make sure that those cells are meant to be there in that body. All right, moving on to multi-gene traits. These are also called polygenic traits. Poly meaning multiple, genic meaning genes. So these are going to be traits that are affected by multiple genes. A good example would be height. Height is a fantastic example of a polygenic trait. Typically, what we see with these polygenic traits is that there's not one trait versus another trait. It's going to be a varying of phenotypes. So that physical display is going to vary depending on those genes. So when it comes to height, think of it this way. You don't have tall and short. You have everywhere in between. So these are going to vary in how the trait actually appears because there's multiple things going into it. You can also think of things like eye color. Like we know what blue eyes looks like. However, there's not just one shade of blue when it comes to blue eyes. You can have that really crystalline blue, a darker blue, kind of a more greenish blue, a gray blue. It just depends. But that is a trait that varies because there's multiple genes that lead to that eye color. There are also behavioral traits that can be influenced by multiple genes. An example is autism. This is a developmental disorder that can be the result of 20, maybe more genes that kind of have this additive effect. So the severity or intensity of the symptoms can really vary widely from one person to the next who have autism. Okay, So kind of going the opposite of a polygenic trait, which is multiple genes leading to one trait, we can also have pleiotropy. This is going to be one gene that influences multiple unrelated traits. The example I have for you is one that we've actually already talked about a few times, and that is sickle cell. Remember that sickle cell is a condition where the blood cells, red blood cells, have an irregular shape. They are that crescent shape. So you can have essentially three flavors of this condition. We can be homozygous normal, homozygous abnormal, or heterozygous, which is going to be a little bit of each cell type, which you can see in those images that are to the left. Now, the traits that this gene affects is the blood shape and the ability to be immune to the disease malaria. Malaria is a condition that you can get from a mosquito bite. Malaria is a parasite, it is a protozoan that ends up burrowing into your red blood cells. It reproduces and then it pops out of your red blood cells. And this can cause anemia. Anemia is where you don't have enough red blood cells to carry oxygen to your tissues. And this can be a very serious problem. You would need blood transfusions as well as to treat the malarial infection. But if you have the genotype homozygous abnormal or heterozygous, meaning you have sickle cell for sure in those two cases, that is the other trait that is affected by this gene. And it does not have anything to do with the shape of the cell. Because remember, with heterozygous, you still have normal shaped red blood cells, but you're still immune to malaria. So two unrelated traits affected by one gene. So, moving on to inheritance patterns that can be kind of weird. So, when it comes to sex-linked traits, remember that this is going to be a trait that is found on the sex chromosome, almost always the X chromosome. Everybody on the planet, every human, has one minimum of one X chromosome. This is very important. You must have an X chromosome. There are no ifs, ands, or buts about it, guys. You have to have one. You inherit this X chromosome typically from your mother. Your father is going to be who determines, not on purpose, mind you, but through the sperm cells, determines whether or not you inherit a second X chromosome or a Y chromosome. Now, because females have two X chromosomes and males have only one, females are going to have a normal genotype involved with their X chromosomes. So you can be homozygous dominant, homozygous recessive, or heterozygous. For males, you are either dominant or recessive. It's not a normal genotype. And it's because you only have one X chromosome, and that is where the allele is going to reside. So, in something like color blindness, which is a trait found on the X chromosome. So, some people kind of think that the sex chromosomes only relate to traits that involve your physical sex. That is not the case. There's a lot of information on the X chromosome that you need to have a functioning human body. Okay, so color blindness is one of those examples of a trait found on the X chromosome, like I said. That means that if you are a biological male and you receive the recessive allele from your mother, you will be colorblind. If you are a female and you receive a color-blind X chromosome from your mother, your father could give you a dominant allele where you won't have color blindness. So what we see with these sex-linked traits is a higher likelihood of males displaying that recessive trait. This is why we call it a sex-linked trait. and they display that unfortunate condition. Usually it's an unfortunate condition. Another example would be baldness. That seems to be linked to the X chromosome, and that is why we see more males going bald than females. Now, is that the only reason people can go bald? No, but it is potentially associated with that X chromosome and why we see males have a different pattern of that trait than females. There are a handful of conditions that can be affected by the environment. An example is identical twins. Now, identical twins start from the same sperm and egg cell. However, in the womb, they are in the same womb, keep that in mind, they will have different conditions that result in their fingerprints being different. Now, if you look at your hand, and you probably can't tell because you'd have to zoom in pretty far to look at your fingerprints, each finger, your thumb to your pointer finger to your middle finger, ring finger, and pinky, are all different prints. If you look at your other hand, your thumb, your pointer finger, your middle finger, your ring finger, and your pinky, they are all different from your other hand. Likewise, you also have toe prints. These are different prints because of different amniotic pressure during the pregnancy of you, essentially. So when it comes to twins, this is different between twins. Their fingerprints will be different. Their genetics are not. Another environmental factor that can produce kind of an unfortunate condition is known as phenylketonuria. Ooh, that is such a hard word to say. We'll call it PKU from now on. PKU is a condition where you have a malfunctioning enzyme that is unable to convert phenylalanine into tyrosine. And when this happens, you end up having a buildup of phenylalanine that can cause toxic levels of phenylalanine that lead to essentially a regression of the nervous system, and especially in the brain. It builds up typically in the brain. What you can see is a slower learning, maybe a regression of speech, that type of thing. It's not a good time. And typically we figure this out when the baby is 3 to 6 months old, if they have this condition. Because how do you get phenylalanine? That is going to be through your diet, and many products will have phenylalanine in it. In fact, a product that has a very high concentration of phenylalanine is diet soda. So much so that a sip of diet soda could lead to essentially death in somebody who has PKU. Now, if you're worried, oh God, Rachel, I drink diet soda. Do I have PKU? Am I going to accidentally hurt myself? No, guys, you would know very early on in life if you had PKU. Okay, other ways that our genotypes can interact with their environment is in something like a Siamese cat or a Himalayan rabbit. Now, they have an odd genotype that allows them to essentially change their fur color based on temperature. So this is why we see color patterns on these types of animals where their faces are darker, their ears are darker, tails are darker, and their paws are darker. It's because the genotype is interacting with that environment, causing the colder areas on the animal to change to a darker color, which is kind of cool. All right. So a trait will not influence the inheritance pattern for another trait, and we actually saw this in lab. We saw it with the corn. The purple or yellow corn did not affect whether or not the corn was round or wrinkled. So this leads us to this assumption here that all traits are inherited independently of other traits. That does not mean they can't be inherited together. We'll talk about that in just a moment. What we call this idea is Mendel's law of independent assortment. And we again saw this in lab in the form of color and seed shape of the corn. We then were able to cross two heterozygous individuals into a dihybrid cross. Now, I'm going to show you this dihybrid cross, but I'm going to tell you a little bit of information that you're going to love here on the next slide. You are not going to see a dihybrid cross on the exam. I like to cover this in lab. I think it makes a lot of sense. I will not make you do a dihybrid cross on the exam. But this is showing us that it doesn't matter what one trait is. They are going to be inherited independently, and that is that law of independent assortment. There are genes that we can see that like to kind of have almost a buddy system where they are inherited together. For example, with our lovely Lindsay Lohan, we have red hair and freckles. These are traits that are commonly inherited together because those genes are located in a similar position on the chromosome. So what we call genes like this are linked genes. Gene linkage ends up leading to, during the process of meiosis, where we have crossing over of the chromosomes occurring in prophase 1, the genes have a tendency to kind of stick together, even though we have that crossing over occurring. That leads to, in the end, the gametes having those genes still stuck together. So they are inherited together. That's why we have things like red hair and freckles being more associated together because they're inherited together. And that, my friends, is the end of chapter 9. If you have any questions, please reach out. I'd be happy to answer those questions. Otherwise, happy studying and make good choices.